welcome to everyone here. I have Usher syndrome. The vision side of it is caused by retinitis pigmentosa, RP. I have passionately supported research for many, many years. And I do so for the obvious reason that without research, there's not going to be any treatment. And I also firmly believe that it is incumbent on me to try to support that research, because if I don't, then how can I expect anybody else to do so? There's a lot going on in the research field, much more than when I first started getting involved about 20 years ago with supporting research. And I think today what we're going to try to do with this panel is to take about 35 minutes, talk with our panelists about a broad overview of the research landscape, talk a little bit about why research takes a long time, but also why things are improving even in that area, and why we should be maybe a little more optimistic today than we were 10 or 15 years ago. Our panelists. I think Bob is to my right. Am I correct?
Yes, you are.
All right, so Bob Bell is the Chief Scientific Officer from Ascidian. And to his right is Ben Shaberman, who is the Vice President of Science Communication at the Foundation Fighting Blindness. Very excited about these two people. They are incredibly knowledgeable. They know how to speak to me as a layperson, and I think it'll be very enlightening to hear what they have to say. So I'll start this off, and by the way, we'll have about 20 minutes of Q&A after we're done. So hold your questions until then. So Bob, I'll start with you. Can you kind of give us a big picture overview of the research landscape in our community?
Sure. Thank you, Martha. Thank you, Martha. I'm happy to, and thank you to the Carroll Center for inviting us to be here. We're really happy to be a part of this community and the event today. I think there's a lot to be hopeful for with what research is doing today, partially based on the knowledge and the technology that continues to advance. We now have a much stronger understanding of the genetics behind inherited retinal disorders. We have a stronger understanding of the mechanisms that contribute to things like age-related vision loss. And that's allowing researchers to apply new approaches to address the concerns or the biology behind some of the causes of vision loss. Speaking from Ascidian's perspective, there's tremendous excitement in the advancement in genetic medicines. The idea of being able to address the fundamental cause behind the loss of vision. That is something that 10, 20 years ago was just not possible. And we now have not just the tools, but the evidence that this can work in people. It does still take time, and I think we're going to talk a little bit more about that. But I think that with the understanding of the fundamental causes of the disease, it allows researchers to really get to the core of the problem and fix it. Right? We want to fix the problem. I think what's important as well is to understand that different causes of vision loss have a different course. Sometimes the onset is different in terms of, is it early in life, middle of life, late in life. In different forms of vision loss, sometimes the cells in the retina, the photoreceptors, the ganglion cells, the retinal pigmented epithelial cells, sometimes they're still there, they're just not working correctly. And in other cases, they're lost. And different treatments need to be considered dependent on where an individual is in the course of the disease, as well as what is going on in the eye. What can we fix? What can we address? And that helps guide researchers in thinking about the approaches that make the most sense for any given person with visual loss.
Great. What I'm going to ask Ben to do is maybe, if I can toss to you, Ben, talk a little bit about, building on what Bob just said, is that we all have different causes of our diseases across the inherited retinal disease space. We have different rates of progression. We are at different stages, some of us early, some of us late. And Bob was just talking about different treatments for these different conditions and different circumstances for each person. Can you talk a little bit about how somebody individually might look at and learn about what might work for them? What might work for me, a senior with no vision left, with RP, versus someone who's maybe a teenager who has a different disease, or maybe the same disease, but totally different stage.
Sure. Thanks, Martha. And I wanted to get back to the hope comment, because there's a fact that I often throw about that I think is really important about hope. And then I'll answer your question. I began with the foundation more than 20 years ago as a science writer. Back then, everything going on in research was in mice. And the foundation had been around for 35 years. And I thought, wow, this stuff is moving slowly. It hasn't moved in humans yet. And I wondered, when would there be change? So without going into like milestones, 20 years later, we now have more than 50 clinical trials underway. We have, if you throw in dry AMD, a few therapies. And there are somewhere in the neighborhood of 50 companies now that are invested in our space. And if you had asked me when I first started, if I ever imagined this day, I would have said, I don't know. We still have a lot of work to do, no doubt, but that's what gives me hope. But to get to your question, Martha, genetic testing, finding the mutated gene causing your disease, is really an important part of the process. And that will help you understand, based on your gene, what emerging therapies might work. And some emerging therapies are targeting specific genes, others are gene agnostic. And another important part of the journey is to get evaluated by an expert, a doctor that understands inherited retinal diseases, so they can help assess where your retina is at, how many rods and cones you have left, how many other cell types. And that can also help you understand what therapies might be relevant to you. We have therapies for people who are emerging therapies, for people who have early stage disease. There are oral therapies that are showing some real promise, some for mid-stage, and then even for folks like Martha, you have pretty advanced disease, there are therapies that are relevant. But I think being proactive about the genetics, the clinical aspect, and following the research, you can really get a sense of what's going to make sense.
Thank you, Ben. To make sure everyone understands, can you explain what you meant by gene agnostic?
Sure, thank you. I get a little nerdy there. Gene agnostic simply means the therapy is designed to work regardless of the mutated gene causing the disease. So as an example, retinitis pigmentosa, there are somewhere in the neighborhood of 80 to 100 genes, each of which, when defective, can be the cause. Well, a gene agnostic therapy for RP is designed to work regardless of what a person's gene is. It could be any of those 100.
Great, thank you. And Bob, we're talking about how people should learn more about their genetics and work with their physicians, their ophthalmologists, retinal specialists. But what are the resources that are best for people to kind of stay engaged and follow the research? How best can someone do that?
Well, I would add, in addition to your doctor, the field of genetic counseling and medical genetics has expanded dramatically. And I really recommend for somebody who has a genetically diagnosed inherited retinal disease to look to speak with the genetic counselor to sort of understand the biology. They really have the specialized training to make sure that folks who might not have a scientific background can really understand what's going on. So I think that's one way. The other is, now, we all know we need to be careful with the internet, but I think there are still some valid and valuable resources, especially those, for example, that are sponsored by, for example, the National Institute of Health or the National Organization of Rare Diseases, some of the more reputable organizations. They can really break down aspects of how the gene mutations are causing disease, and that can help folks understand, for example, what they can expect in terms of progression, how long a thing is going to take. The other thing I would mention, and this might be a little bit harder, but to always ask questions, and no question isn't worth asking in this space to learn more. Scientists like myself are always willing to talk about biology. They may not have the answer, they may not have the solution to say, here's what you need to do to fix it, but most will be very willing to just speak about what is known and what's not known. And I think in my own experience in having those conversations, it's a really fruitful discussion and helping people to sort of understand what's going on in their circumstance.
Now, this may be a good spot, Ben, to kind of explain a little bit the genetic testing and counseling services that are available. Could you just give a broad overview of that?
Sure. That's something else that has evolved enormously over the past 10 to 15 years. Back in the day, nobody knew what their mutated gene was because genetic testing was not accessible. But different academic research centers have genetic testing available, and those panels have gotten very powerful and robust. And I'm also proud to say that the Foundation Fighting Blindness, we offer a no-cost genetic testing program that really any eye doctor in the U.S. can order along with your health, and that can include genetic counseling as well. And Bob, you articulated the benefit of genetic counseling. The genetic counselor will help you understand the results of the genetic test, whether they're conclusive, inconclusive, maybe other people in your family should be tested, or that's recommended. The genetic counselor will help you with that. And then along with that, we launched, it's been more than a decade ago, we launched a patient registry called the My Retina Tracker Patient Registry. And now whenever you're genetically tested through our program, you're automatically in this registry. And it's safe, it's secure, we do not ever give out personal information. But researchers and companies can go in and look for potential candidates for clinical trials. They don't get your personal information. If your record happens to match their interest, we at the foundation, we have 2 or 3 people that are authorized to contact you and say, you know, Mr. Jones, company XYZ is running a trial you appear to be a potential candidate, then it's up to Mr. Jones to contact the company. So that's a very powerful resource as well. And then I'd be remiss if I didn't mention the Foundation Fighting Blindness website as a great resource to just learn about what research is underway and the diseases as well. That's how I get my paycheck, is by working on that stuff, so.
Yeah, it is a great resource, no question about it. So, I want to ask another question. You know, I often get, you kind of spoke to it a little bit, Ben and Bob as well, how 20 years ago you didn't think you'd be in the position you are today, or that we are in general in terms of the research. But many times I hear from people that are just not motivated to support research or even pay much attention to it because they may have at one time, but enough time elapsed without anything happening, they just lost interest, and that was that. So it's worth, I think, talking a little bit about why it takes a long time to do this sort of research, to get something in the clinic. We had Luxturna, something for LCA, particular genetic RPE65 gene mutation for LCA that was approved back in 2017, and I think a lot of people thought, oh, we're going to start seeing stuff coming into the clinic, and here we are. There has not been another approval for an inherited retinal disease, although there have been a few for AMD. So what is it that takes the time? What are the barriers, aside from funding, what are the barriers that have been in place that you think maybe is crumbling a little bit now? What is your thought about time? And I'll start with you, Bob.
Yeah, I think the example with Luxturna is a great one, and it really has been a trailblazing program for other inherited retinal disease gene therapies. And so every gene therapy requires its own design. We often call it the construct, but you can think of that as the drug. Right? And that's something that takes quite a bit of iteration to make sure you're expressing and restoring the exact appropriate healthy gene and RNA and protein that you want in the correct cells, right? So figuring that out is important. And it takes researchers time to test different constructs in models such as cells in a petri dish, in animal models such as rodent models that might also have the disease. And then importantly, you want to make sure that the drug is well tolerated, and that often requires studies in larger animal species. Oftentimes in the ophthalmology field, this could be in things like non-human primates. And those studies can take years. 6 months at minimum in terms of looking at the tolerability, but then also to make sure that you have a persistent effect of the drug. So getting the construct right, running the appropriate studies that gives the researchers the confidence that you can deliver the drug to the right cells, that it expresses the right amount of the RNA and the protein, and that it shows some therapeutic benefit. All of that happens before even the larger safety studies. The other aspect of drug development is what we call, often termed CMC, right? And it really is, it stands for, you don't need to worry about what it stands for, but it's about manufacturing the drug, right? And this also takes time, and it's appropriate that it takes time because you want to make sure that you can manufacture the medicine in a very consistent and high quality way. So once you've identified you have the right construct, it works in the animal models, it seems like it's safe in the animals, before you can treat humans, and this is really in a lot of alignment with regulatory bodies like the FDA here in the US, you need to make sure that it can be manufactured in a very high quality, high controlled, reproducible ways. typically at minimum, that type of campaign is gonna take a year and a half. That just is what it is. You have to make multiple batches, characterize them, test them. So then once you've passed all of that, you say, okay, I'm ready, I have alignment with the regulatory agencies such as the FDA here in the US, you start what's typically called a phase 1/2 clinical trial. And the primary goal here is safety again, right? The primary goal for your first time in patients is not necessarily to instantly correct the disease. You're trying to make sure you go in cautiously, conservatively, and ensure you're doing no harm. That requires exploring multiple different levels of how much drug you get and waiting time in between each individual who gets the drug to really ensure that the medicine is safe. You know, the preclinical animal studies gives you that confidence, but at the end of the day, until you're in people, you don't know. So I think all of that is years, Martha, just to really get the confidence it's safe. Of course, you know, if I think about Ascidian, right, we're in a phase 1/2 trial for Stargardt disease. Right? Our primary goal is safety. We're exploring 3 different dose levels. So we start at the lowest dose, we treat a few patients, we have an independent, unaffiliated data safety monitoring committee that reviews all the results and then gives a recommendation for us to go to the next dose level and to the next dose level. And so again, that takes time. During those safety assessments though, of course we're looking for benefit. Of course we are, right? And again, depending on what type of disease folks are treating, that could be a functional improvement. You could see an improvement, which is the case for Luxturna. Or in other diseases, you might be looking to slow progression, comparing to things like, what do we know about the natural history of the disease? Comparing to, for example, oftentimes in a phase 1/2, you only treat one eye, so you can compare to the other eye. But again, these things can take time. Once you finish that, then you typically would need to move into what's called a phase 3 study. Oftentimes, these are controlled, where you have a much larger population going in at maybe 1 or 2 different dose levels. And here you're confident the drug is safe, and now you're looking for the benefit to get something approved. And again, that can take a few years on its own. And along the way, these studies are very expensive. Right? I'm not going to try to sugarcoat that. And so identifying mechanisms to fund the study is a big part of what we do in biotech. Right? And there are a lot of great investors. Ascidian has been very lucky to be backed by a VC firm called Appletree, right? But it's challenging to raise the funding and to find investors who have the patience for you to deliver to patients, right? And I think that's also a part of the journey as well. So I know that was a lot there, but I'll pause.
Yeah, so, and all of that is so, it is incredible. It does tell you why it does take long. Ben, do you have anything to add to that? And maybe if there are any thoughts about whether there's anything changing today based on our experience and what we've learned that might be helping to improve clinical trials, maybe even speed them up a little bit, anything like that?
Sure, and Bob, you did a great overview of why the research takes so long. And I wanted to provide a little case study that also answers your question, Martha. So there was a company, I'm thinking it was 7 or 8 years ago, called ProCure, a company in the Netherlands, and they had 2 RNA therapies. One was for a form of LCA, another was for USH2A actually. And their early clinical trials, the drugs performed pretty well. In fact, the LCA drug performed well enough that they moved it into a phase 3, the last phase. It was a small trial because a lot of these inherited retinal diseases are rare. It's hard to recruit patients. Anyway, in that trial for the LCA therapy, the drug was called, or is called cepofarsen. It worked for some people pretty dramatically, but ultimately they did not meet the endpoint that they agreed upon with the FDA. And if you don't meet your primary endpoint, then you're kind of in trouble. And when ProCure didn't meet that endpoint, they ran into the business decision: do we even stay in ophthalmology? Because they would have had to have launched another trial. And they decided to drop out of ophthalmology. And at the time they said, we're going to find a partner for our assets. And I'll be honest, I was not optimistic, because I thought there's no way anybody's going to pick these up. It's a tricky road. But then a company in Europe called Thea, Laboratoire Thea, that's my French for the day. They picked up both assets. They spun off a division called Sepal Bio, and both those drugs are back in clinical trials now. And it's miraculous. And what they did is they changed the design of the trials. And I won't go into the details, but the way they have the control versus the treatment group, the way they're recruiting patients, they learned from that first miss. And with any trial that doesn't work, even if the drug doesn't cross the finish line, we learn. And I think Sepal Bio now, they got smart, and now that they're learning better how to design trials. And one last thing that I'll mention, we at the foundation and a lot of companies are working more closely with the FDA on trial design. And we're actually trying to negotiate with the FDA to be a little more flexible as well. So we've learned a lot, and we're being more proactive about how we design trials so they're more likely to succeed.
And I'll make a quick comment on that, Ben. The working with FDA is, FDA has certain, as Ben said, endpoints, but not always most meaningful to us. And what we are trying to do as a community, the foundation, companies, other organizations, is to convince FDA that there are other endpoints that are very meaningful to us, and that they should be considered. So if one trial doesn't get you so many lines on an eye chart, which is what it was supposed to do, but it gets you able to see a crosswalk because you have better contrast ability, so you start seeing shapes, so you know you're not going to walk into a tree right there. That's valuable. And so that's a lot of the discussion that's going on at FDA right now by various companies. Okay, we probably got about 5 to 10 minutes here before we move into questions. I want to ask, I guess it's a double-folded question, Bob and Ben. And first is, we talked a little bit earlier about why should we be more hopeful today than we were 10, 15 years ago. And in answering that, I'd like your thoughts on what you are most excited about coming up in the pipeline.
Sure. I think there are 2 or 3 things I'm very excited about. And one is the advent of therapies for people with the most advanced vision loss. And I'm thinking about the optogenetic approaches. It was maybe 10, 12 years ago that optogenetics kind of got on the radar screen. And in a relatively short amount of time, there are now 5 clinical trials underway for optogenetics, and there's a company that has actually filed an application with the FDA to get approval. Now, and these are approaches for people who have really no rods or cones left. So I think that is very exciting. And on the flip side, we're seeing a lot of promise for those gene agnostic approaches that are designed to slow disease progression. They could be oral medications or other approaches. And those are performing well in clinical trials as well. So I think we've just gotten a lot smarter. We've learned from mistakes, we've learned to work better with regulators. And I also think the financial tide has turned a little bit. When the pandemic hit and inflation went up, simultaneously, things got really tough for the biomedical space. But I think we're seeing a reversal of that. We're seeing more investment. And because we're smarter, I think companies are more excited about investing in our space.
Bob, what are your thoughts?
I agree with what everything Ben said. I also, I'm very hopeful for the gene-specific approaches as well. I think, you take with Ascidian's program, the drug's called ACDN01, we are correcting the ABCA4 RNA in patients that is causing the disease. And by correcting that the RNA, we fundamentally believe we can have radical efficacy. Now, when the disease has progressed, we're hopeful that we can stop it in its tracks, at least slow it down. But you can also imagine a world where if we can identify patients early and we have confidence in the safety and the efficacy, you can imagine a world where you're treating before symptoms are really onset. And you've now corrected that underlying genetic cause. And I'm hopeful for a world where for Stargardt's and other inherited retinal diseases, we're able to treat such that folks never even have to experience the initial vision loss. I think that's going to take time, but I truly do believe that is in the future. The other thing that I think is important is, oftentimes biotech companies, large pharma, have commercial targets, right? They want to go after indications where there are a lot of patients and a lot of potential for revenue. That's the sort of standard business model. And what happens then is some of the more ultra rare diseases frankly get left behind. And I'm seeing a lot of creative business thinking that is changing that mindset. You're seeing organizations working with folks like FFB, other non-for-profits, and other rare diseases that aren't necessarily even related to the eye, but are creating a path, a framework, if you will, a playbook for addressing more ultra-rare conditions. And I'm really hopeful for that because the technology is here, right? The technology's here today to be trying to tackle some of these things. And I think it's important to sort of move beyond the mindset of, well, we have to have a peak year sales of X in order for us to want to work on that. I think that will still exist in a lot of the sectors, but I'm, again, very hopeful for a lot of the creative thinking that is happening in the field that's going to allow folks to go after some of the more ultra-rare genetic disorders that traditionally have been a little bit left behind.
Okay, thank you, Ben and Bob. And I think what we'd like to do right now is to open the floor to questions. I think there's somebody walking around.
Yeah, hold on one second.
Hello, my name is Brian Cola. I'm congenitally blind. I had congenital cataracts. Had my astigmatism and no intraocular lens. The question I have is, I was also working on possibly new kinds of glasses, kind of like the bioptics, but that would allow for someday the possibility of someone like me to be able to drive a car.
can start. So I can say, so that is not something that, you know, myself or my company is directly working on. I think that there is a lot of effort in wearable devices. I think, you know, I remember recently, I think at, it may have been an FFP sponsored event, a lot of excitement about even the new Ray-Ban glasses. This is not a plug for Ray-Ban. I don't get paid. If they want to send me free sunglasses, that's fine. But I think there is progress being made there. In terms of a pair of glasses that would allow you to drive a car, I think that is going to be challenging for a number of reasons. This is just my own opinion, but I think that, you know, the amount of technology going into wearable devices to help improve people's life in terms of even being able to enter a room and having the glasses tell you what is in the room. I think some folks have started to do that, or to help you sort of read. So I think there is innovation happening in some wearables that is exciting, and look forward to seeing the progress of that.
Yeah, I think what you said about is spot on. I guess the other thing that I'll add, and I will not claim to know a lot about what's happening in the front of the eye where you're more affected, but we're getting a lot better at growing new cells in tissue. And for somebody who has, you know, serious damage or lack of development in a certain part of the eye, I think that's hopeful. I don't study that part, so I don't know what's going on, but I know you can get new corneas, and they can use stem cells to grow new corneas, and I know they're trying that for not only the retina, but other parts of the eye. And I think in the long run, that provides real hope. The other comment I'll make is a project we're working on, actually through government funding, an agency called ARPA-H. I won't talk about ARPA-H so much, but it's whole eye transplant. And the goal of the project is to successfully transplant a human eye in about 5 to 6 years. And there's, I think our team alone that we're part of, there's more than $40 million going into that project. We call it a moonshot because it's very ambitious, but we're excited, and we're going to learn so much from it regardless. So it's that kind of effort that I think provides hope for some of these really challenging situations.
Hi, I have 2 questions. First, I'll save the questions and you can answer them in order if you like. Stem cell therapy, it seemed years ago it was kind of a hot topic, and is it my imagination or has it died down? So I'd love to hear what you both think about stem cell therapies and where it's at. And second, I'd love your opinion on cord blood. So I, of course, saved mine because I had this disease when I had children. There were a number of us who had eye disease, and we decided when we had children to save our cord blood, and here I am 23 years later, I'm still paying fees and such. And I do have a lot of colleagues that are young females that have an IRD, and they wonder, should they do this when they have children? So I know this is maybe a little off topic, but I would be really interested to hear what you have to say about that as well.
I think, I mean, Ben just mentioned the stem, so the first question related to stem cells, has it died down? I think it's probably coming in waves, but I think it's back. I think, you know, the biggest challenge with stem cells is folks have figured out how to take a stem cell, in addition, turn it into having the characteristics you want for it once it's in the body. I think there's been a tremendous amount of progress there. Now the trick is, how do you get it to graft? How do you get it to actually, in for example, the retina graft? And I think there's been a lot of innovation in the sense that we know you can't just inject them. They need to have more of a, you know, biomaterial that they're engrafted to. The other thing that is challenging with the stem cells is the idea of whether or not it's kind of to your question about the second question, whether or not you need your own stem cells or there can be a pool of stem cells that could be used for everyone. And I think ultimately for a scalable and a product, a cell-based product that can reach the masses, you really are, folks are hoping you can do it so it's what's called a non-autonomous, meaning that there's sort of a bank of just general cells that you can do. So I think there are folks who are very actively working on this. There are clinical trials on stem cell, adjacent based approaches for inherited retinal diseases, and we'll have to see how they progress over the years. I think that the question on the second question is I don't feel the right person to answer that. I think it's a much more of a personal decision on doing that. But I think there's just always hope that there can be a sort of breakthrough and having access to all the tools in your toolbox you can is an important consideration.
So Laura, the question you asked about cord blood, I've gotten that question many, many times, and do I personally know the answer? No, but I go back to the experts, and generally speaking, they don't see a benefit in it. And I'm talking about people like David Gamm at the University of Wisconsin-Madison. And mentioning David, he's been working on stem cell therapies his whole career, 15, 20 years. And just so people know, the way we're making new rods and cones now is by taking a small sample of blood or skin, genetically altering those cells so they revert back to a stem cell-like state, and then coaxing them forward to become new rods and cones. And that's what David's therapy, now in a clinical trial through a company called Blue Rock, that's what that is. And you have a couple of choices about where you get the blood. You can have it be the patient's own blood, and in that case, it's the same genetic profile. You don't have to worry much, or you worry little about rejection, but it's also going to have the same genetic defect. So those blood cells will have the RP gene. Or you can find people in the public whose immune systems are more favorable to not rejecting, or their cells not being rejected. And it's the latter approach that is being used in the Blue Rock trial. So, as Bob said, it's kind of time intensive and expensive to have these personal cell therapies. But if you can find the donors who have the favorable immune profiles, that's a better way to reach the masses. But I'll echo, the big challenge has been integration and grafting. And if there's any person on the planet who can figure it out, it's David Gamm at the University of Wisconsin.
Oh.
Is there any effective treatment now that slows or stops the progression of RP?
Not in a gene agnostic way. Well, I'll let Ben answer first.
I'm sort of hedging.
Okay, go ahead.
So, a few weeks ago, we reported on a trial in Australia for a oral medication called N-acetylcysteine amide, or NACA. And long story short, it's derived from an FDA-approved drug called NAC. It is modified, so it is not an approved drug. But in that trial of 49 people in Australia, it was a phase 1-2 trial, it slowed the rate of photoreceptor loss by 50%. It's a small trial, and a third of the people in that trial didn't get the drug. They were the control group. So we have to be a little careful.
Was it gene specific?
No, it's gene agnostic. It's just something you take orally. It's a powerful antioxidant. So oxidative stress is a big problem, especially in RP. And so now this company, Nacuity, is moving toward a phase 3. They're talking to regulators. Now the original drug that it's based on, called NAC, that's also in a large clinical trial in the US at like dozens of sites. More than 400 people are enrolled. And we'll know if that earlier version NAC works in probably 2 years. They're hoping sooner, but we were saying, why do some trials take so long? Well, if you're trying to slow progression, you need a lot of time to see if progression is actually being slowed. Because these conditions, they may not seem like they're moving slow to you who are affected, but they do move relatively slowly from a clinical standpoint. So I wouldn't have answered that way a few weeks ago, but it's an exciting development, and fingers crossed.
Do we have time for one more?
We have
One
time
more.
for one more.
My name is Louis, and I've tried to take part to clinical trials against the macular AMD and GA. And I was told every time that since I was on a trial some years ago, that my eyes were injected, or one of the eyes was injected, I was not a candidate. Could you clarify why is this happening?
I think there's probably a distinction between being a candidate for a new clinical trial versus potentially being a candidate for what hopefully is an approved drug. Oftentimes, in early clinical development, if you have been treated with another type of therapy where the new company cannot determine whether or not that previous drug that you received could still be having an effect, and not just an effect on a benefit, but could it have a safety effect? And so if the idea for a new company starting a new clinical trial, you don't want to confound the results with variables that may not have to do with the drug you're developing, but potentially due to something that someone had received prior in their life. Again, I think that's for early clinical development, especially your sort of phase 1/2s where you're assessing safety. I think as a program develops and there's more confidence in the benefit of the drug, I think some of those limitations on inclusion criteria, which is what you're referring to, start to go away. But I think they start to go away once either the drug is approved or there's just a significantly higher degree of confidence in the effect that it can have. But that's the primary reason, is that folks want to make sure if you're trying to assess a new medicine, you're not confounding those results with something that could have, that doesn't have to do with the drug being tested. I hope that answers the question. I'm happy
Just
to follow up on that.
conceivably after 10, 12 years, it might still be in the body, in the eye.
I think it depends, sir, on what exactly it was, and I'm happy to chat with you sort of after to give you some further thoughts. Yeah.
Thank you.
Okay. I think we will stop here so we can transition to the next panel. I want to thank Bob and Ben for an excellent discussion. Really appreciate your thoughts and feedback, your feedback on where you think things are going. I think it's a really, really exciting time. I do encourage everybody to really pay attention to the research, try to support it if you can. There is a huge amount going on right now. Ben mentioned almost 50 clinical trials right now. Doesn't even count the ones that are in the pipeline along the way. So thank you very much, and let's have a round of applause for them.